Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Daniel Geschwind, who's the Senior Associate Dean and Associate Vice Chancellor of Precision Health at UCLA. He has been at the forefront of research in neurogenetics and in autism spectrum disorder in particular for a long time. I had the pleasure, actually, of collaborating with Dr. Geschwind during my PhD, and so I've read a lot of his work, and I'm really excited to have you on the podcast today.
Oh, thank you so much. It's my pleasure. It's great to be here.
You've spent most of your career in research, obviously, but you had a kind of interesting start, actually. You spent a couple of years as a management consultant at Boston Consulting Group, and I wanted to start there because that is not something that you get very commonly. You sometimes get the reverse where people go into management consulting later, but how did you end up taking that path, and what made you decide actually you wanted to go into research after that?
Yeah, some of it is very personal in that I had a family of scientists and academic physicians, but scientists. My dad was a physicist who was the chair of the solid state physics department at Bell Labs, super academic, even though it was industry, it really wasn't industry. It was, and so I had this notion that the only route in life is science. My mom was an artist, and she just wanted us to do something that wasn't very stressful, that was nurturing. And so, I went to college and I majored in chemistry and psychology. I was really interested in the brain, and I liked chemistry a lot. I wasn't quite going to go the route of physics that my dad went. It was very intimidating. So chemistry was the closest thing, which I really enjoyed. I loved chemistry. And I went to Dartmouth, and it's a fantastic chemistry department for undergrads. So that was part of it too. Just the faculty and stuff were so nurturing and excited about their stuff. So anyway, so I got into that, and then I was looking around for jobs, and I got a teaching job at a private school in, one of the teaching chemistry. And then I just started to interview for, with the companies that were on campus, like JP Morgan and Morgan Stanley and, Bain and BCG. And I went and I interviewed. Bain was a really small company that had just started. It was a spinoff of BCG. And so but BCG I really loved because Boston Consulting Group, because it was very, very academic. The people there were really unbelievably smart and driven, and so it seemed like an exciting thing to do. And so I went and learned a ton, met a lot of very interesting people there. But I guess when I was looking, at least from that vantage point at that time in my life, when I was looking at careers and thinking about things, something stuck in my head, which was that I really wanted to have a significant life. I don't know what that means. At the time, I didn't have a family or anything like that. Of course, that is probably the thing that makes you most significant, your family. But, I wanted to have impact, and I wanted to work on something that kept me engaged. And what I saw in business in general is that most people worked to retire at age 51, 52, and that seemed like a long way away when I was 2 years old. It seemed like a long way away. It seemed like an entire life, but I wanted something that would engage me and kind of keep me interested, and so I realized that even though I had been moving away from my kind of family business, you could say, that that I was probably more suited to that. And And so I applied to MD, PhD programs because I really wanted to take care of people and do something, again, that would have impact on people. So it seemed that the combination of doing biomedical research afforded by an MD, PhD would be, the way to go. So, it was a process, really. I have to say, I really enjoyed my time at the Boston Consulting Group, even though it was short. It was 2 years. I was a research associate, but I really got a lot out of that. But, I realized when I was 50, the way I looked at it was when I was 50, looking back on my life, is that what I wanted, how I wanted to spend it? For many people, it's an incredible opportunity, right? Because it was a wonderful place to work with wonderful people. But I realized I wanted something that would keep me, as long as I lived, kind of engaged in it.
You also spent quite a bit of time producing, filming, starring in ski movies. I thought that was really interesting.
Oh, it wasn't a lot of time, actually, but I spent 1 year when I went to college. My first year, it was a shock to me for a lot of reasons. And again, I won't bore you with the details, but I decided my sophomore year to take the year off. And so I went to work with a Dartmouth alum who ran a ski filming company called Cine Ski in France. And I worked for him, and we made a ski movie with Jean-Claude Killy, Henri Duvillard. Henri Duvillard was the current world pro champion, an amazing skier, and Jean-Claude Killy is the icon of every ski racer. He's the only person to have won all the medals in an Olympics. Of course, there were only 3, I think, in that 68 Olympics. But yeah, so it was amazing. Again, that was fun. But when I came back, I was, I'd worked really hard, and, I loved to ski. And so I got to really ski a lot and work hard. But I really came back kind of knowing what I wanted to do at that point. That really helped me think about, it was interesting. And so I got into chemistry and started to work towards, I thought I wanted to be a doctor at that point. But then I started to have second thoughts when I looked at my family and felt like somebody in my family should make some money, right? And so I should go to business. So I should go into business and give it a try. So that, I kind of, it was a circuitous path.
You spent a huge amount of your career working on autism. I want to start there. What did we know when you initially started out in the field, and what are the big things that have surprised you that we've learned over the last couple of decades as that's unfolded?
Yeah, I think so. When I started working on it, it was because of a relationship with a family here who had an autistic son, friends. And they founded this organization called Cure Autism Now. And I really didn't know much about autism at that point, but I was trained as a developmental neurobiologist and as a neurologist, and I felt I could have some impact there. So I started to work. It was clear to me that what we knew at that point was that there was a strong genetic component. There had been twin studies published by Sir Michael Rutter and others that had really shown a very, very strong genetic component. And so I worked with Cure Autism Now to build a gene bank called AGREE, the Autism Genetic Resource Exchange, with the idea that it wouldn't be my thing. It would be something for the field to build the field. And it was essentially a biobank with clinical material and assessments attached to it. And the idea was, if we build it, people will come. People would want to work on this. And that's indeed what happened. Hundreds of laboratories used that. And it really, I believe, opened the modern era of autism genetics in many ways. Subsequently, the Simons Foundation, which had a lot more resources, built even larger biobanks that have also had, I'd say even a larger impact on autism research. But the idea was, let's make samples available. When we started, we thought, and so the other thing that we did is we collected families, multiplex families, with the idea that this is highly heritable, and therefore we should enrich for those with 2 or more children with autism. I still believe that's a really strong approach. It's based in fundamental kind of genetics. You have a highly heritable condition, you should enrich for heritable factors. So, so that's how it kind of got started. And I wasn't really doing autism research per se in my lab. I was, running this as, with this private foundation. And then we decided, NIH was interested in helping to support us. So I wrote grant to NIMH, which included some science with some other colleagues who were very, very generous. And we ended up, and that kind of slowly brought me into autism research. We funded the biobank partially via NIMH funding. So it was a private public kind of partnership. And we began to build autism genetics in that way. And it's, so one of the things that interested me was thinking about autism not as a thing at the time. This is in 1999, right? But as now this kind of thinking I think is standard, but at the time it wasn't, as a kind of quantitative trait, just like height or weight or blood sugar or anything. So the idea was you have social cognition. There are people like Bill Clinton who were on one end of the social cognition, who are amazing geniuses at this, and others, we all fall somewhere. And so if you measured the components of autism, the psychological cognitive components, you could do quantitative trait mapping. And so that's what I started with, really looking at language, which I was very fundamentally interested in. That's what I kind of started my career with, looking at development of fetal human brain asymmetries that are related to language, and genes related to the development of language-related cerebral cortex. And so that's really what we started with, and nobody was really taking that approach at the time. And even still, people kind of poo-poo it, and I still think it's a powerful approach. Quantitative trait mapping is more powerful than using kind of qualitative categorical traits.
And so what do we know today about the particular genes or gene networks that are involved in this continuum that you described, or the spectrum? Language is part of it. What else is?
Well, it's interesting because a few years ago in DSM-5, language was taken out, and it was just put as a something that you characterize autism, you characterize language, and you report it as part of that, just as you would report intellectual disability. But the core features are now the repetitive behavior component and the social component. And so we've actually done some work recently which suggests that the polygenic risk score for autism broadly actually loads onto language features and language development as well. And we published that, in 2023 in PNAS, and now we validated that in larger cohorts. So I think that language is a fundamental component of autism, even though it's not in the current rubric. And that's one of the difficulties with studying a psychiatric disorder, in that, there isn't necessarily a pathology or something to hang your hat on, right? Like in stroke or multiple sclerosis or even in dementia, where you can find protein aggregation. And so that was one of the things that kind of captured my attention. One of the things that we did that I think in the long run will change the field, I'm not sure it has really yet, is we decided to try to define molecular pathologies in psychiatric disorders, with the notion that, as the microscopes, as our ability to see more and more with higher and higher resolution has increased, our ability to categorize and to understand diseases in general has improved. So you have, stroke, which you can see often in the tissue, and multiple sclerosis, but you have other things like Alzheimer's disease, where you might see some atrophy of brain regions, but what really defines it is the cellular pathology, and you could only really see that once you had microscopes. Well, so we reasoned, now having the ability with microarrays, and more recently with RNA sequencing, to measure the molecular state of the tissue, that maybe there'd be a molecular state that defined the pathology of disease. And so we tried that in autism. Irina Voinigo, who was a very brilliant and adventurous postdoc in the lab, decided to take that on. And with a relatively small cohort, we had under 20 cases in controls, so under 40 samples total when we came to the end of it after all the QC. And she was still able to identify a very strong pattern that distinguished autism from controls that was present in, let's call it, between 2/3 and 3/4 of cases. So that kind of molecular pathology has been an anchor in the way that I think of the disorder. And we've gone on then to use that. If you think about it, the transcriptome or epigenome is a quantitative trait, and it's a phenotype. And so we've used that as a quantitative phenotype to compare across psychiatric disorders, to begin to understand what's distinct about them and what's shared, because there also is shared genetic, even between things as kind of disparate as autism and major depression and eating disorders. There is shared genetic risk. So most of the risk isn't, but there is a shared genetic risk. So kind of studying on the one end, you could kind of almost think about it as reverse genetics, right? Starting to define the molecular consequences in the state of the tissue. Then on the other end, we've been working to identify genes and try to connect those things and understand how genetic variation impacts the brain. I guess as a neurologist, I was trained as a developmental neurobiologist originally, which has given me a great biological foundation. Of course, medical school does as well. So I feel like I really come at this as a biologist who's trying to understand mechanisms. And what I realized during neurology training was that so much of what we were seeing had a genetic basis. And that by finding the genetic basis, it gave you a causal anchor that could really anchor your studies. So instead of doing an MRI image or even a transcriptome, all of these things are phenotypes. You really don't have known causality within that. You can't assume that they're causal. But when you find a genetic variant that's associated with a disease, you can, be pretty certain that it's causal. The disease didn't cause the variant. And so, that kind of a light bulb went off in my head, especially when it came to these more mysterious disorders like autism. So, 80% heritability, really just was ripe for genetic studies. One just needed to find enough patients and families. And so, interestingly, just to go back a little bit with Agree, when we started Agree, one of the other things we did, we realized that there were studies going on that were trying to ascertain families with autism. But they were taking place at academic medical centers. So the families would have to come in, they'd have to be ascertained. And that's a huge barrier when you have a disabled child, or two of them, or three of them. And so we went out to the families. We literally flew around the country. And when I say we, not me, actually, we had phlebotomists. We actually hired a pediatric neurologist, Sarah Spence, who got her teeth, really sharpened her teeth in Agree, saw hundreds of autistic children in the period of just a few years doing a fellowship. And she recently retired, but she had been running the clinical autism program at Boston Children's Hospital for many years now. So yeah, so that's kind of how that went, and that enabled us to really recruit a lot more and ascertain a lot more families much more quickly. It was expensive though, but it worked.
A common question that people have is around the relationship between autism that presents in, say, a child with intellectual disability and some kind of syndrome, and the more, I guess, autism in an adult that may have a job and be a little bit more high functioning. I don't know if there's a term or set of terminology that you use, but should we think of those as two separate examples, or are they both part of the same continuum in some way from a molecular biology perspective?
So I'm going to say both.
That's fair. That's a fair answer. We have time to unpack it.
Yeah. I'm going to say both because just like, you could think of other things, like if we just talked about cardiology for a second, or cardiometabolic disease, right? If you take the top 2 to 5% of polygenic risk scores, those folks have Mendelian-like risk, just as if they had a mutation in a predisposing gene. So you can think of, autism is very interesting because it's an early onset childhood disorder. And so what we know now is that even though about 80% of it is heritable, or it has 80% heritability population-wide, that about 15% of kids with autism, or people who we assess, will have a major single gene mutation, usually not inherited, usually de novo in most cases. And that kind of recognition and that model led to identifying families with simplex autism. In other words, now you don't want to have a family history. You want kind of a bolt of lightning. and like Down syndrome, a de novo mutation that is in the sperm or egg and not inherited from the parent. And early on, there were statements made that, oh, this is going to be 50% of autism. It's going to be de novo. This is going to be the majority. And I kept asking people who were saying that, very, very smart people, you could say, why? I mean, if there's 70 or 80% heritability, how can that be?
Right.
Right?
Not enough de
That's
novo
not
mutations per generation, probably, right, to do that.
right. And then the other issue was, if you look at the twin, I mean, there's just too much evidence that that wasn't the case. But it turned out that that's low-hanging fruit. If you have a parent-child trio, you can sequence them and identify a de novo mutation much easier than you can lower effect size heritable mutations. And it's basically because of the effect size. So the field really moved to that. And so most of what is known about the genetics of autism in terms of genes that cause autism is based on de novo mutations. But there's several things that one needs to know about that. Number one, they're rare, obviously, and they're not common. So that no single mutation or gene or let's say no gene accounts for more than 1% of autism.
Right.
So that part of autism, that 15% plus or minus, that's just an estimate. That's de novo, is a collection of rare disorders, hundreds of them. And each one of them is kind of own medical syndrome in a way, right? And so that's been tractable. People have found a lot of mutations. The difficulty is, though, number one, you want, to find enough patients with that particular mutation and understand what it's actually doing, right, is not easy. And then when you make an animal model or a cell model, you have another difficulty, which is another challenge, which is that most of these mutations, even those with very high penetrance for autism, also cause other things. And I know you're well aware of this. There basically are broad causes of developmental disorders. Now, they cause intellectual disability, epileptic encephalopathies, autism, plus or minus, right? Broad developmental delay. And so the question is, when you make a mutation and you're seeing phenotypes in a model, what is that phenotype related to? Is it related to the intellectual disability? Is it related to some, obviously, most of these are developmental mutations. And so the model is that these mutations don't cause autism. These mutations disrupt brain development at the midpoint, early and mid fetal development, setting development off track in a way that predisposes to a number of neurodevelopmental conditions, including autism. And some of these are rather specific for autism, but that's the exception rather than the rule. Most are pleiotropic. And so that challenge then becomes, what you really need to do is to study a lot of mutations. You can't just study one. Well, you can. You'll get to some mechanistic understanding of that gene's function, and you'll understand that syndrome. The question is, does that help? Is that necessarily autism, or is that SCN2A? It's SCN2A, and that's really important, right? Don't get me wrong, super important. And so that is actually going to lead, very, very similar to the what's happened in cancer. You have a specific gene, it's a specific mutation, specific pathways, and then you can develop drugs to that. And so one of the big questions in autism that's been on my mind since I wrote an article in 2006 or 7 about autism, many genes, kind of common pathways question, is there convergence like cancer, right? Cancer converges in proliferation and invasion. Every major cancer type either, hyperproliferates and invades the body, and that's why it's malignant. And, this is malignant cancer. And so, what are the features, of autism at a molecular and cellular level, and maybe at a circuit level, if we're talking about brain disorders? And so I think that's a really challenging but central question in the field. At the same time, Maybe we don't have to understand that to treat some of these disorders, right? If you can figure out what's going on in SCN2A mutations or Dravet syndrome, or some of these, especially the epilepsies are a little bit, maybe more tractable, you can come up with gene therapies and other approaches.
Yeah, what is your current thinking on that question? Is there evidence of some kind of convergence that could be treated with a higher order approach, or is it going to be around gene therapies and targeted approaches to the hundreds or maybe thousands of different subtypes?
I think the jury is out. I mean, it's interesting in cancer that convergence was identified early before the molecular era. And so we applied things that were focused on invasion and proliferation. You remove the tumor so it can't invade, or you debulk it, and then you treat it with radiation or antimitotic agents, which prevent it from dividing but have toxic side effects. So it may kind of be like that we'll find some areas of convergence. It's going to be quite different because it's unlikely to be cellular. Some will be, but the big convergence is going to be kind of circuit. So, through neuromodulation and stuff like that, will we be able to, kind of treat, or help people with autism, with some of their comorbidities? It's an interesting issue. I think right now, what's very exciting to me is that for these conditions, I do want to make a point though, because you brought it up about autism as a spectrum, right? You have, because it's now considered a spectrum, you have people with these mutations who have a medical condition, right? It's a medical genetic condition that is really severe and would benefit from medical intervention. And then you have people who are very high functioning who just need accommodation. They don't need to change. Nobody wants them to change who they are or anything. We need as a society to accommodate them. Yeah. And so, that's created some misunderstanding, I think, of kind of where, how people like me and others are thinking about this, right? In no way are we trying to cure somebody who doesn't want to be cured. It really doesn't need to be cured. They just need to, to have accommodations made to make their life, better, right? But like in terms of these folks with major mutations who are, severe, really disabled in many ways and can't enter normal life, the advent of nucleic acid therapeutics is very, very exciting because most of the mutations that cause autism, but not all, are haploinsufficient in that they knock out one copy of a gene. And there are those that are dominant negative, right? Certainly. And some of them that we think are haploinsufficient are likely dominant negative, but that hasn't been identified. But we actually can figure that out. One of the ways we can figure it out is using modern methods such as CRISPR activation to turn the non-mutant copy of the gene back on to some reasonable level. One of the tricks is that these genes are very dosage sensitive in general, so that overexpression of them is thought to be as detrimental as underexpression, but there are ways to get around that. I'm happy to talk about that if you wanted to. But
yeah, please.
Yeah, but I think, just there are now people targeting different ways, right? Like you could target the 3', you could target RNA degradation, right? And so you could increase the stability and half-life of the RNAs so that goes up. You could use CRISPR-A to target the promoter or enhancer to turn the gene back on, et cetera. And that is going to turn out to be much easier than actually providing the gene itself. And you don't need to because there's another copy. Obviously, for recessive disorders, a protein or now RNA therapeutics turn out to be extremely promising from my perspective. And now we have delivery systems that can deliver RNA therapeutics. So that's very exciting. So on the haploinsufficient end, one of our approaches has been to, rather than targeting promoters, which are often not that tissue specific and not stage specific and open in many cells, so you could get off target effects. If you turn the gene back on, you're turning it on in cells that you might not want it in. We are working on targeting enhancers, which is trickier and probably on average less effective, but still perhaps safer, because if you can identify an enhancer that turns a gene back on, that's going to work when that enhancer is open in that cell, in the right cell at the right time is the idea. And so we've been working on that and have some success. So I'm very bullish on nucleic acid therapeutics in general, biologics, because, in many ways their safety is going to be much more understood, right? Like biologics have, certain safety issues, and nucleic acid therapeutics do as well, depending on how they're delivered. But that's a kind of generic issue. It's not going to be different for each one, most likely. Obviously, there are going to be exceptions to that rule. So I think it's a whole new it's a whole new ballgame in a way, and I think this is going to lead to potentially even in utero therapeutics down the road. I don't think that's going to happen quickly. I think there are a lot of regulatory and technical issues that have to be overcome, but I actually do see that as a promising part of the future. It won't surprise me by the time that I'm retired that we're, treating people with these devastating medical genetic conditions in utero.
And is that because some of the evidence is pointing to where the brain development is starting to break down is even going back that early?
Yeah, most of these genes, if you look at the de novo mutations, they converge at mid-gestation.
Wow.
In certain kinds of neurons, although some are broadly expressed. So it's not like there's one, answer here. Again, if you think about what autism is, again, backing up, it's a syndrome, it's a behavioral syndrome. So there's no reason to expect there's going to be one cause. There's no single biomarker for autism. It's, there are a lot of ways to get social dysfunction and difficulty with mental flexibility and repetitive behavior, right? And so that's what we're seeing. That never surprised me. What always surprised me is how people thought with 100 or 200 families, we were going to identify a gene for autism, or 5 genes or 10 genes. That never made sense. It's kind of like cancer in a way, or infections. It's just such a broad syndrome that so, and in fact, I don't know if you have children but for those of us who have kids, if you think of social cognition and the mental flexibility, like not doing repetitive behaviors and having some flexibility around behavior, that's a lot of what happens between 0 and 3. You add in language, which is, I think, another core component, and sensory motor integration, which is also disrupted in at least 90% of folks with autism to some degree. You end up with a lot of development being impacted. The only thing that really helps autism, where it really stands out, is in the lack of intellectual disability and in most people who have autism. It's just these rare 15% or so of rare haploinsufficient de novo mutations, or whether they're recessive or haploinsufficient, but the rare genetic forms, those are strongly associated with intellectual disability, motor delays, and other comorbidities, even multi-organ system involvement. I once wrote a review for, I think it was Annual Review of Internal Medicine, where I just looked at the syndromes associated with autism, and 60 or 70% of them had cardiac abnormalities associated with them. So again, consistent with these being kind of medical genetic syndromes that are multi-system, impacting development early enough. So, but interesting question. How much can you reverse if you had a 7-year-old or a teenager?
Yeah, that's what I was going to ask.
Yeah, really unknown, but the work in mice in several syndromes from, several genetic syndromes associated with autism suggests that there is some reversibility. The most famous one is Rett syndrome. Now, Rett syndrome is not autism, and it's not even an autism spectrum. It's its own thing. It was originally, but it really is very, very different. But it is a archetypal neurodevelopmental disorder that has tragic neurodegeneration and deterioration associated with it. And that gives you a sense that you could intervene, because the child develops normally up to a point postnatally and then deteriorates. And that deterioration goes on, in some cases for more than a decade. So maybe there it's not quite as surprising that you can reverse many things in an animal model. The jury's out on many of these kind of early fetal expressed things. One of the more common of the rare mutations is in CHD8, a chromatin modifier, and that's expressed very, very early brain development, goes down. So the question is, if you expressed it, in an adult or in somebody postnatally, would that be good? Number one, or would that cause problems? But number two, would it actually reverse some of the findings? And people are working on that using animal models, and that's an exciting thing to look at. But there are probably a dozen different genetic mutations for which there's evidence that turning the gene back on in an adult is beneficial. May not be a complete rescue, not surprisingly, but it's very beneficial. So that holds some hope, even for people who are living with these mutations now.
Yeah, I saw results from Regeneron's trial in genetic hearing loss relatively recently, and they saw effect in restoring hearing and even up to 16 years old, which this is a kind of syndromic hearing loss. Yes. And I was pretty amazed, obviously different than autism or developmental disorders, but I'm really curious across the board of what the therapeutic window is going to be for gene therapies, and in some cases it may be very surprising.
Yeah, I mean, obviously, if a receptor or a signaling molecule is missing, even if it had a developmental role, of course, most things do, and you put it back, the signaling should normalize it, as long as the circuits are there. The interesting thing, too, will be not just coupling these gene therapies with, not just giving them, but you're going to have to couple them with training and learning, right? I'm sure even in hearing, right, it doesn't just bring, you have to train the ear to hear again. So if we give genes back, I mean, this is another complication, I think, to clinical trials, is that I once heard a parent talk about this in a very eloquent way at a meeting, and the parent is a physician, so, they're very sophisticated, biomedically. And so what the parent said was, imagine that you can't read, you're dyslexic, right? And there's a gene, right? So, or a medicine you can give. Think about the medicine as turning the light on or off. So the light's off, you can sit next to the patient and be training them to read, but they can't read because they can't see the page. You turn on the light, you can't just throw the book in front of them and say, read. You have to train them. So it's the combination of turning on the light and the training. And I think that's a brilliant metaphor, that if you're 9 years old or 10 or 20, and you've lived a life that, where you're perceiving the world differently, and all of a sudden a light is increased or turned on a little bit, you're also going to need to have the proper training. Yeah. And so when you run a clinical trial, how do you incorporate that into the clinical trial? Because it's the combination of the training plus the therapy going to lead to the largest gains. And so, that creates a lot of challenge for clinical trials all across the board, as well as regulatory issues as well. In other words, is that drug now only approved alongside that that behavioral therapy or whatever it is? So, yeah.
I want to talk a little bit about your commitment to data sharing and all the work that you've done. So obviously we talked about Agree. You've also been involved with Simons and Psych and Code. And I think recently you've started Autism Genetics and Human Diversity program that's focused around getting African-American families more represented in this kind of research. I'd love to hear a little bit about what you think we need to do as a field to take a big leap forward in autism genetics or psychiatric genetics more broadly, because that you've been at this for a really long time and have been at the core of some of these major data sharing programs.
Yeah, no, it's a really, really great point. I mean, it's an interesting thing. As a neuroscientist, again, when I got into autism genetics in the late 90s and started to think about it with Cure Autism now, it seemed to me that medical genetics, and I'm a neurologist, so at that point, the spinocerebellar ataxias, Rett syndrome, and other things were being tackled, and these monogenic conditions. Yep. And it seemed to me that people's competitive advantage, and this is borrowing my term from Boston Consulting Group, the competitive advantage in human genetics at that time was what you had in your freezer and could hide from other people. Honestly, I mean, I hate to say that maybe that is, of course, I'm not meaning to discount how difficult it was to find genes, and you have to be very clever. There's some brilliant, like finding the Duchenne muscular dystrophy gene is just like by cDNA subtraction and stuff. I mean, that's a brilliant, study. So like there's sparkles of brilliance throughout all of this, right? People using innovative techniques to kind of find genes. But a lot of the folks would find that, it's linkage, brute force, sequencing of the region, backs and, brute force thing. But you couldn't do it unless you had the families. And the people who had the families were the only people who were going to find the genes. And it was usually like this race, to see who was going to do it. And that to me seemed, number one, not in the patient's best interest, not in the field's best interest. And, maybe, just not the right way to do science. And so when we started to agree, the whole idea was it wasn't going to be our biobank at UCLA. It was going to be open from from day one to get the field going and really a proof of principle that something like this could really work and change a field, and that it wouldn't have people tripping all over each, the idea is, well, if you share, then you're going to have people tripping all over each other doing the same thing. And it turned out not to be the case, really. Same thing with the Simon simplex collection. Those things have revolutionized the field, those kind of sharing databases. Agree was more shared. Simon's was a little bit more locked down and more, a group of people were working on it who had access to it. Agree was really much more wide open, but still Simon's had that same spirit, the idea of let's build it and people can use it. And so, that's really the Simon's resources have been extraordinary for the field. So you were asking, basically, how I feel about it. I mean, I think I just told you, I think that one of the things that's been missing, and the Human Genome Project really nails that, is having a map, right? Like when you want to go navigating and discovering things, having maps makes it a lot easier. Pre-mapped, it's harder. And so one is the map of the genome, but that's really not a functional map of the genome. And being a biologist, I've been fundamentally interested in functional mapping. So we started very early on to look at gene expression and gene regulation, chromatin maps. And so now there are these incredible projects, mostly funded by the Genetics Institute, right? The Human Genome Institute, to like the 4D genome, looking at gene regulation and stuff. But in the brain, one is going to need tissue and stage specific maps of how genes are turned on and off and function essentially. And so we've been working on that. And then you can ask how function is dysregulated in disease. So you also profile disease brains as well as part of that. And that's what PsycEncode really was. It's kind of in its last legs. My sense is it was incredibly successful, and I'm surprised that it's kind of ended as a program. There still are a few groups who are funded, but my role in that is as chair and stuff is I'm no longer doing that because our grant is over and there's no longer a PsycEncode program to be involved in. Those types of programs, right? From PsycEncode to UK Biobank, right? Like you create resources that then people can use. It saves decades and hundreds of millions of dollars if everybody had to do their own little profiling to get somewhere, right? So now people are telling me, we use this data to look at XYZ. Well, that data took us 5 years to collect, but it allowed them to do something in a day.
A couple of weeks, yeah.
Or a few weeks. So that's the idea, is to build these maps and these resources so that things that would normally take years to do now can be done in hours or days. And the investment in those things is extraordinary. And in fact, in diseases that pharma is very interested in, like Parkinson's and Alzheimer's, they've recognized that and put a lot of money along with NIH to create like AMP-AD and other programs, right? And so there is a recognition that these resources are broadly useful. So I think data sharing is is really an essential part of things. And, it really hasn't sunk into neuroscience. Everybody wants to do everything their own way. I do my physiology this way, you do it that way. We can't really share. And I think, the basis of science is reproducibility. And so, it's certainly gotten better and better over time. It's easier to share genetic data than it is to share images and to share physiology, but it is shareable. So now I'm part of this SciGene program. That's another NIMH program that's really set up a database that connects genetic mutations in stem cell models all the way through morphology and physiology.
Oh, interesting.
Which is a real challenge. And, we'll see how all of that goes, but it's working. And it's it's kind of the next step in things. Like I view, now that you have 100 or 200 genes, what do you do? If you have, what do you do with polygenicity, et cetera? You need a lot of samples and you need to study a lot. You can't just study a single gene. And so my lab has begun to focus on high throughput biology, just the same way that genetics is a high throughput method approach in and of itself, to actually do phenotyping in high throughput in a cellular and molecular way and physiologic way. And so building, leveraging robotics and all kinds of other technological innovations to be able to do things that were usually done by one person at a microscope over a day to do thousands, So we've been working on that, and SciGene kind of embraces that. It's really about developing high throughput phenotyping assays and data sharing. Yeah, so that's really been the key, and setting up these databases. And and so you were asking me where things are going. I mentioned UK Biobank as one example. I think so. Part of the future is high throughput biology to allow geneticists to connect genetic risk to actual biological phenotypes. And things like iPSC models for many, many tissues, different tissues allow you to do that in 3D now. So that's very exciting. But the other side of it is the leveraging of our biomedical infrastructure and the electronic medical record. And you mentioned at the beginning, one of the hats I wear is directing our precision health efforts at UCLA. And one of the things that got me very excited about that is my belief that rather than funding collection of tens of thousands of cases, we can get tens of thousands of cases through the biomedical record. People would say, the critique of that is, well, they're not well phenotyped, blah, 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 it's not good enough. And it's true, you want to be able to go back and contact people. But what's really surprised me as somebody who believes fundamentally in the importance of rigorous phenotyping at every level is how usable the electronic health records are with regard to phenotyping. For example, in our UCLA Health Biobank, where we've genotyped now about 130 or 140,000 patients, we can look and see, do we replicate known associations that are done in carefully, curated, GWAS studies? And the answer is yes. Every single one. There's not a one. If we have the sample size of European patients, we're able, and that's another problem, is that most of this has been done in European patients. And so we're lucky in Los Angeles in that our biobank is about 40% non-European. And that's leading to many, many discoveries, and really interesting. And so I think having diverse populations isn't about DEI, it's about biomedicine for everybody. And Zach Cohane had a brilliant paper in New England Journal of Medicine almost 10 years ago now, where they showed that rare variants that were thought to be causal for certain cardiac diseases were not rare if you looked in African American populations, and they turned out to be benign. So even if you only care, like our current administration does, about people of European origin, having the diverse populations enables you to understand what's going on in those populations. So having, of course, that's not our point of view. Our point of view is really health equity and taking care of everybody in our populations. And to do that, we need a diverse biobank. And that's what also drove me more than a decade ago with my colleague John Constantino and Ami Klin to start working in African Americans because I realized all the gene discovery, at that point when we started, we'd only identified 10, 15 de novo mutations instead of the 200 that have been identified now. But I could see, it was just going to grow almost linearly with sample sizes up to a point.
Yeah.
And so, okay, people are doing that. I don't need to do that. They're doing that. That's going to get done. That's not that interesting. What's interesting now is increasing the diversity and going into African American or Hispanic populations like Joe Buxbaum and his group are doing, where we focus on African American for population genetics reasons, really, right? Everybody's out of Africa. They have the highest frequency of rare variants. And so not de novo, but rare inherited variation. So we thought that would be a really worthwhile population to study. And of course, they're massively understudied in research in psychiatric diseases in this country and across the world. So it was a no-brainer in a way. So that's been very exciting. We're working on our first paper, which I think is going to be very interesting. Like we thought we'd find some things that were different, and we have. Surprisingly, like sometimes it surprises me when what we thought was, possibly correct.
Right. Yeah.
So,
the rare example of not a.
Yeah. Usually we have a hypothesis that it's like when we started profiling autism brain, I thought we were going to find different subgroups. That it would break into, that we wouldn't find this convergence at the level of transcriptomes. So, yeah, it's interesting. Yeah. So, one thing I know we're running out of time. One thing we didn't talk about much, but which has also been a huge kind of passion and focus of mine, has been trying to organize like analytic methods that help you organize transcriptomes. So these network methods and stuff. And I would say from a neuroscience standpoint, I feel like that's our biggest contribution to the field, is showing how network, very simple co-expression, correlation can kind of lead, simplify and lead to better analyses. And, my hope is that smarter people than me will come up with better methods and, keep so that eventually we'll have a theoretical foundation to biology that all these analytic methods are based on. Right? In genetics, a lot of the theoretical foundation is evolution.
Yeah.
And that helps drive things. But like when we're looking at transcriptomes and stuff, and we start looking at biological phenotypes, that's often ignored.
Yeah, I never thought about that. Who is working on that, that theoretical foundation that you're talking about?
Oh, I don't know. But like I've always felt that network biology is a kind of theoretical foundation in a way. And so if you can identify and so when Barabasi and others and my colleague Steve Horvath came up with these methods that we worked on with Steve to apply to biology and showed, like Mike Oldham, who was a grad student in the lab, we profiled the human brain, or we literally got other people's data, and we showed that the transcriptome had a structure. And at that point, like I thought that was really profound, because people think of the genome as having a structure, DNA, proteins have a structure, the cell is like a factory, but RNA was like just gobbledygook. And so showing that there's a structure to how things work. And so I think somebody you might want to, there are people working. I'm not sure if people are working on it from the standpoint of trying to get to another theory, but I think I do think that Barabasi's work showing the kind of scale-free topology, or kind of hierarchical scale-free, it's kind of mixed scale-free and other things. I think that was a major breakthrough, honestly, in kind of thinking about how things relate to each other. Otherwise, biology is just descriptive. Whatever we're doing, even if you're measuring the biophysics of a channel, you're describing what's happening. It doesn't fit into some enormously large theoretical notion of how the brain works or how the organ works and stuff. I mean, it fits into smaller things about the nerve and its conduction and the way channels ions movement. It fits into that part of physics, but at a larger level, there's not that higher level thinking. And so I do think that some of this network biology, I mean, it's interesting how one moves from kind of, where the revolution will really occur, I believe, is moving from description to, kind of a fundamental theoretical understanding, but maybe that's asking too much. I mean, already we know that our understanding of mechanisms, even though they're not grounded in some theoretical construct, is sufficient to lead to therapies, which means that we do have some, the kind of necessary level of understanding at some levels, mostly outside of the brain, right? In cancer and immunology and other areas. And the brain remains this area where it's tough.
Someone who this reminds me, I'm going to check out more of Barabassi's work and follow up on that. But it's also reminding me a little bit of Jonathan Pritchard in the GWAS world. He does a very good job, I think, of there's a huge amount of observation in the field, but then actually trying to wrestle with that and bring it into something that's more theoretical.
Totally. Yes. Thinking in a really big way. And it's interesting because the kind of, what's it called, omnigenic hypothesis, although, one can criticize it, and we have, for many reasons, because most of the disorders actually look polygenic, really, but one has to do that. And that's the whole idea of, and so, it takes a certain kind of brilliance to do that. And I'm really grateful for that. I had one student whose almost entire PhD thesis was based on creating brain networks from GTEx and looking at it from every angle to see, do transcriptional networks or protein interaction networks or transcription factor regulatory networks, all three of those, do any of those clearly obey a omnigenic hypothesis as stated? And we weren't, it looked more polygenic, but, we've done some other work to look at that as well. But I think there's some fundamental observations in omnigenic, such as the cell type specificity of disease.
Yeah. That
was a fundamental observation that is really key. And so there's a lot of truth in some of the stuff, and whether, and of course it doesn't spell out a lot of details, so you have difficulty, but there is a lot of truth in it. And again, I think you hit the nail on the head. There aren't a lot of people who are kind of, operating at that level in the field. It's interesting. That's, and maybe genetics is a little bit easier than neurobiology in that way, and in that we have a sense of how, it is interesting though, because that omnigenic hypothesis really sets forward a path for trying to understand how genetic variation is actually operating. Because I was going to say, we have a sense of how things work, and we really don't.
Well, I think it is. I think you're probably right that it's easier, just because you alluded to this fact of the reverse causation not being so much of an issue in genetics, right? What came first, but when you start to think about transcriptional networks and brain biology, there's a whole lot of opportunity for things to influence other things in circular direction. So you have more complexity to deal with, right, than you do in a genetic model where you can make some simplifying assumptions.
That's really true too. Yes, and we also, and the genome is knowable.
Yeah,
to some degree, right? To a first approximation, it's like measurable, and all of our genomes are now measurable, and so it's a constrained space that in which we're operating. Yeah, it's a really good point.
Thank you, Dan. This was a ton of fun. I learned a lot. You've been doing some amazing work. It was really cool to hear about what the next frontier is for your lab as well. So look forward to continue to keep track of your work. And thanks for spending an hour with us today.
Thank you for listening to all of my stuff. And I look forward to staying in touch.
All right. Thanks, everybody. And we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.